Aiyetan. Good afternoon, everyone, and welcome to our third FAJIM Scholars Mentorship session for 2026. Dr. Paul, we are very delighted to have you with us today. Thank you for making the time to engage with our scholars. This is one session we've particularly been looking forward to because your journey across medicine, research, leadership, and innovation is a very strong example of the kind of possibilities we want our scholars to see, learn, and begin to imagine for themselves. So thank you. To our scholars, I encourage you to make the most of the opportunity, listen attentively, engage with the conversation, and ask the questions that matter to you. You're welcome to share questions or comments in the chat as the session progresses. And the attendance link will also be shared there, so please keep an eye out for it. Okay. So we're also trying to become more intentional about creating opportunities for our scholars to develop their communication and leadership skills. And in that spirit, today's session will be anchored by our FAJIM scholars. Our anchors today are Toiba Ige, Muhammadu Kabir, and Adidami Laregani. Thank you to all three of you for volunteering to moderate. And we hope to see even more scholars step forward for these opportunities in future sessions. Okay, so with that, I'll hand over to Toiba, who is on the call, yes, who will take us through the session from here. Toiba, over to you. Thank you.
Okay, thank you so much, Ms. Mariam. Good evening, everyone. First off, I'd love to know if I'm audible. Am I audible, ma? Hello, everyone. Am I audible?
Yes.
Yes, you are.
Okay, thank you
Yes,
so
you are.
much.
Thank you.
Thank you so much, ma. So good evening, everyone. My name is Toiba Ige, and I am a fourth year medical student at Lautech. And it is my esteemed honor to welcome us all to the third FAJIM Scholarship Mentorship session for this year. This evening, we are going to be learning from someone important, a very important figure. We are going to be learning from his insights and his knowledge as he takes us through this topic. Looking to the horizon, hello, the future starts now. So, without further ado, I'd love to hand over to my fellow scholar, Muhammad Kabir, as he takes us through the bio of our speaker for this evening's session, Dr. Paul Aiyetan. So over to you, Muhammad. Thank you so much.
Okay, assalamu alaikum. Thank you, Taiba. Good evening, everyone. Hope I'm audible.
Yes, you are. Good evening.
Yes, you are.
Okay, good evening. My name is Muhammad Kabir, a FAJIM scholar and a 300 level MBBS student from Katsina. I'm very delighted, sir, and honored to recite this professional bio of yours. And it reads like Dr. Aiyetan is the founder and CEO of NEOPATHOLOGY Corp, a biotechnology research and clinical diagnostic development company. Prior to establishing NEOPATHOLOGY Corporation, Dr. Aiyetan led high-performing teams at Interben Biosciences in Silicon Valley, California, the United States Frederick National Laboratory for Cancer Research, National Cancer Institute in Frederick, Maryland, the Institute for Biomedical Research in Massillville, New Jersey, and at the Johns Hopkins University School of Medicine in Baltimore, Maryland. He received his medical training and degree from College of Medicine, University of Ibadan, Nigeria. A jointly offered Master of Science degree from the Johns Hopkins University, Zambia, Krieger School of Arts and Sciences, and the Wharton School of Engineering, and his Doctor of Philosophy degree from the George Mason University School of Systems Biology. Funded by the U.S. National Institute of Health, NIH, National Cancer Institute, and the National Heart, Lung, and Blood Institute, NHLBI. He was a postdoctoral fellow at the Johns Hopkins University School of Medicine, Department of Pathology, Center for Biomedical Discovery and Translation. At the Johns Hopkins University School of Medicine, Dr. Aiyetan was an investigator on the NIH/NCI-funded Clinical Proteomics Tumor Analysis Consortium, that is CPTAC, and the NHLBI-funded Programs of Excellence, PEG, in glycobiology initiatives. Besides being at the helms of affairs, he led NEOPATHOLOGY Corp's research, development, and translational efforts. Dr. Aiyetan has several research publications in highly respected scientific journals, such as Nature Biotechnology, Nature Communication and Cell, including US patent filings and applications. So I'm delighted and honored to recite this bio of yours, and I hope my FAJIM scholars, my fellow FAJIM scholars, will all listen to our presenter of today, Dr. Paul Aiyetan. Thank you, everyone.
Thank you. Thank you. Appreciate it. I guess it's my session to go ahead with the presentation, right?
Yes, please, Dr. Paul.
Okay.
Share your screen. Oh, thank you.
Thank you very much. Thank you for the warm introduction. Thank you, Professor Abdulkarim. I really appreciate you giving me the privilege to reach out and talk to our esteemed callers here today. And thank you, Ms. Miriam, for coordinating and facilitating this meeting. And thank you to everyone that showed up today to come and listen to these amazing talk. I hope you find something really exciting to take from here today. And feel free to stop me at any time if you have any questions and clarifications. I will gladly welcome it and be patient enough to respond to you as we go on. Okay, I will share my screen in a moment. And please let me know if you can see my screen.
Okay.
Can you all see my screen?
It's coming up.
Okay. When it's up, you let me know so I can get started.
Yeah, clear.
Okay. Thank you so much. So today we're looking at looking to the future and how the future starts now. And for me, that topic was kind of like spontaneous to me when Ms. Miriam asked me what would be the topic for today. And I thought it was something that would take you through my journey through medical school, through my career development and to where we are right now as medical professionals and where we see the future going, and we have a conversation from that. I'll start off with this quote from Dr. John Shaw. It says that the future is not some place we are going. And oftentimes we think, oh, when I get there, when I become this, when I become that. But one we are creating, as in the future is what we're creating. The path to not to be found is the path to the future. It's just something you start looking for, and you want to find. That's something you have to make. Well, and John Shaw is an American, is a professor and a political theorist. And if you read the bigger text of that particular quote, of where he picked up that quote, or where we picked up that quote, well, it talks about the future is something that is created instantaneously the moment you take a step. And the person journeying into the future actually gets changed by every step they take. And so I hope I do justice to actually kind of like shed more light on how that has been for me, and hope we can have a conversation from there. So my path actually started from something from clinical curiosity that flowed my scientific inquiries from the University of Ibadan. And I'll say I try to imagine a little while back, but before I go ahead, let me ask this question. Miss Miriam, how many minutes do I have?
You have about from 20 to 25 minutes.
20 to 25 minutes. So I'll try to keep myself within that time slot.
Thank you.
My 200 level, I took a course called Culture and Civilizations. And for the first time, I think my perspective of what is scientific actually got challenged. It's so easy to think it's only physics, physics, chemistry, and biology are the only scientific fields of endeavors of humanity. But then in that particular course, I was challenged and began to realize that those are not only the scientific fields, and science in itself at wrongly being assumed and defined wrongly. And so because of the wrong definition of what sciences was, we kind of assumed those were the only scientific fields. I got to know anthropology was a scientific field. I got to know that even philosophy was a scientific field. And that's what at the topmost end of the scientific journey, you get a doctor of philosophy, because essentially you're philosophizing, you're actually imagining, questioning things. And so going forward to medical field and medicine, I started to question why some things happen. And oftentimes you will see, and I don't know if it's common these days, the medical text will say something is idiosyncratic, as in something we can't understand why it happens, but we know it happens because of the nature of the patient, about something that is really about the patient. And still in the University of Ibadan, right, I started to ask that, what if we can know more about what is wrong with the patient, for the information we can derive from the patient with respect to the test we perform, and now we answer the test, and how many molecules we can see at a time. And towards the end of my medical school was when the Human Genome Project was completed, and next generation sequencing were becoming a thing, microarrays were becoming a thing, and newer technologies that made it possible for us to see deeper and profile more broadly what's going on with the patient at a time, but become commonplace. I got so excited about it. I said, perhaps by knowing this, we can actually tailor treatments and diagnostics with respect to what exactly is wrong with patients. And perhaps we can answer those questions of idiosyncratic process about, we don't know why this happens, but we perhaps could know better about what is happening. So I saw a postgraduate program at the Johns Hopkins and in the UK, and I got accepted in the Johns Hopkins University. The fascinating thing about my journey into Johns Hopkins was that Johns Hopkins has a kind of a program where you can take classes anywhere across the Johns Hopkins Institutes, whether it was the School of Engineering, School of Arts and Sciences, the School of Medicine. But what propels you is a question you're trying to solve, and what you think you can get the knowledge that will help you answer the next question that you want to answer. It could be in engineering, it could be in arts and sciences. And I was going into a field where there was no curriculum for. And I thought I was living medicine. A friend of mine asked me when I was going to Johns Hopkins, you go into School of Engineering at the Johns Hopkins University, are you leaving medicine? And my response back to him was, I do not see a wall separating medicine from engineering. And I do not see a wall separating engineering from philosophy. I think over time, because of human endeavors and specializations, we erected walls that are not supposed to be. And so we don't understand the entire body of knowledge of what we ought to know, or a entire body of knowledge that helps us address what we ought to know. And I began to realize that the question I'm asking is not something I will only get from the medical textbooks, but something engineering probably solves, whether in biophysics or whether in computational gardens or some engineering field that I don't see medical field that will help me better understand what I'm trying to learn. I'm trying to take multiple amount of information that the Human Genome Project produced. Mass proteomics was becoming a common thing where we can actually look at multiple proteins in a reaction and process at the same time. And also next generation sequencing was getting a whole lot. How do we understand this? How do we make sense out of this? How is the data even formed? How does the data changes from one form to the another? Or where does the knowledge come from? What is knowledge and what is useful in the knowledge I generate and I see? And so there were questions that really floated in my mind. And I thought, I need an engineering specialty to actually better understand this. And Hopkins allowed that, a whole lot of that. And still asking the same question took me to my PhD program at the George Mason School of Systems Biology, where they were starting to have a more structured form of learning where you can begin to look at the human body as a system, and you could build a twin nature or structure of how things do work at the genetic level to the protein level to how protein modification builds into an entire picture to understand disease processes. And luckily for me, I got support from the National Cancer Institute and the National Institute of Health at the Johns Hopkins, started from the Johns Hopkins University onto the Frederick National Lab, where I was actually stationed in a lab where I had all the resources I needed to do the screening and questioning. And so my journey from asking the question of how can we better understand what is going on with the patients and be able to tailor-make their own treatment based on what we know is unique about the disease processes, that's the diagnosis, and to better derive good outcomes, to get better outcome or better outcomes in managing them. And so in a nutshell, that's gonna be my journey. But where did I start from? And the world I was going into, this was the field back then. We thought we had a lot of information. We thought it was becoming more and more easy, it was becoming more and more easy to actually profit the entire genome. And this graph on it, can you see my cursor, everybody?
Yes, we can.
Yeah. And these here shows you the cost of sequencing the entire genome at a point where the Human Genome Project was completed, where we now know, well, supposedly had a first draft of the entire genetic makeup of the human being. This was the cost back then, but it was soon seen that the cost was becoming more and more cheaper to actually get that information. And so there was an explosion in genome sequencing projects, as in what makes up the genetic, what is the genetic makeup of maybe a honeybee, genetic makeup of, see the Yoruba genome even here. I know a professor from the College of Medicine in Ibadan that was also part of this project here, the Yoruba genome here. The cancer genomes, as in like specific kind of cancers. And so we thought we were gaining a whole lot of information. And this is the world I came into. And this is what actually made me question, can we use this amount of information to better derive tailor-made treatments and the diagnosis for what is going on with the patients? But here is what we have now. Much more than what we anticipated at the time I was getting out into the field that we have. So if we had a hurricane, as at a time I was getting into the field here, we have now, it's a tsunami of information. Now we not only are seeing the genome, but now we can see protein. And so we can see the entire protein and see the protein that actually constitute the makeup of a particular person in a disease state, within a disease process, what the finest accuracy that you can actually, that we've never seen before. And beyond that, we can actually also see the metabolomes and the metabolites that were derived, that were synthesized on given biological processes. And not only are we beginning to see that, we can actually see the entire tissue microenvironment and see the relationship between what is happening within the cell and what is happening around the cell. And there's something fascinating along my journey. From my transition from Johns Hopkins University to the Frederick National Lab, well, we had started to look at what we call the epithelial molecular signatures in prostate cancers. And then the technology had been invented called microdissections, as in where you can actually microdissect a tissue to the cellular level, where you can tease the epithelial layer of that tumor and the stromal layer of that tumor and sequence those two layers. And what we had begun to see was that we were seeing some crosstalk happening between what we see in the epithelial signatures and the stroma signatures. And this was 2009, 2010. And we were beginning to see evidences that the cancer of itself, for many years, we had thought about it was something that originated in the cell. Yes, that's where it's a molecular process that's well established. But the entire initiation process and the progression and ultimately the outcome of the disease process, it's not solely dependent on that molecular change and process. Like if you go back 10 or 20 years before then, you see penetrance and variance. I've been trying to explain such a situation where you see people with the same molecular changes, but having different clinical outcome or clinical progression and states of the disease. And then we're seeing for the first time evidences of molecular signatures and crosstalks between stroma and epithelial structure and cellular processes. And so began to wonder and imagine that the way we were looking at molecular biology is probably not optimal way, where we take out the cells, we digest the cells, extract the DNA, digest the DNA, try to sequence the DNA, put that together, understand the molecular changes. And maybe that's not the optimal way to look at molecular processes and changes in the tissue microenvironment. Maybe we need to study these processes and these molecular processes in situ, where we can see the cell in which these changes are occurring. We can see every cell within the same microenvironment, and we can see how they talk and relate to one another. And this was the beginning of what we call the spatial biology. And then a new technology came in. I wouldn't go into those details, but we see that changes across time. We moved into pantomics, where we can actually look at the cell and the tissue in situ. And as I said, we started to see at the beginning of 2008, 2009, where it was possible to do single cell RNA sequencing, where we can take a single cell out and actually sequence that cell and tell what exactly is going inside that cell. Contrary to the way we had done it prior to that, where we take the entire tissue and digest the whole thing, called bulk DNA sequencing. Not only are we able to see deeper into the tissues and the tissue microenvironment, we can also image better. And this is just MRIs become a thing, and there's something called FAJIM, as in this is similar to electron microscopy, where we can actually look from the very, very teeny tiny molecular process to the 10 to the -9 resolution of what is happening within the tissue and create a structural morphological change and look at the morphological changes in that. All of these are created an avalanche and a tsunami of information we need to piece together and understand biology. Remember, we started from saying, if we can know what is happening in the cell and then as for molecular processes, we would know how to treat the patients. But where we're today, we're not only looking at what is happening in the genome anymore, we're looking well far and way much more deeper than that. We're looking at the proteins, we're looking at the cell processes, we're looking at the imaging processes. And so we're looking now in the future, what we're looking at in the present and into the future going, we'll see a situation where we have a multi-omic and multimodal approach of looking at disease process. And you can see within the short period of time of my training and learning, spanning this short period of time since I left medical school, we have transitioned from what we had in the late 20th century, where we are dealing with syndromic processes and we are observing and doing stethoscope and x-rays and labs. So what we're looking at right now, we're looking at better MRI and imaging approaches, and what can actually characterize the entire genome. You can actually have your whole genome sequenced even now within 7 days, have it returned back to you. Now we're curing diseases and trying to cure diseases because we now have a deeper understanding of molecular processes. And now we think we can cure diseases. But to the horizon, we now have ability to do even greater than what we've ever done before. So we're moving into multi-omics and multimodal approaches where we're actually seeing better signals and better outcomes in the way we actually can treat patients, not only just treat, but also prevent diseases. And so the problem we see though right now is that the problem that we emphasize never did go away, but approaches to it changes from time to time. We thought we had a hurricane of diseases process. We had a hurricane of data. Now we have a tsunami of data. We thought we had showers of data. Now we have an avalanche of data that we have to piece through our physicians and clinicians and biomedical scientists to better understand this process. So we thought we could see better by just imagining that if we could sequence the entire genome, we could cure diseases. Remember, that was the future we had in our mind. And we took steps to get into that future. But the future we have created and the moment we have created is one greater and far much more complex than what we actually thought we were seeing and where we were going. I'll get back to that a little bit. So today we're seeing a shift from broad spectrum, one size fits all treatments to highly targeted specific patient molecular therapies, where targeted therapies are becoming a mainstay in treating patients with cancer. and even immunotherapies where you actually can program the person's immune cells to actually attack the cancer as ways of treating the patients for cancers. I was seeing an approach where we actually started to study diseases not just as a small entity, but as a system at a system level, where we're looking at the entire organism, because we do understand that diseases are complex. They're very interconnected. It's not just an epithelial disease. It's not just a stroma disease. We see the epithelium and the stroma are actually talking together and cross-talking with events that are happening in the liver and immune systems, and all the entire endocrinological system of a particular patient actually responding back to a particular disease process in one tissue. So it's not just simple isolated signaling pathways, but an interconnected complex biological networks. They're actually part of the disease process that we need to understand. And this is not possible to the human eye, and this is not possible just to the ordinary human being. We need a system we need machines to help us. And quickly we do realize that classical approach, and this is the way I said it when I started my journey in the precision medicine space, classical approaches would not cut it given the amount of information we have. But we need an approach where we can actually, like what machines to help us see beyond or go beyond, we can actually reason. The most intelligent human beings can only reason in the second to the third dimensions. But with machines, we can reason into the 1,000th dimension. And being able to reason to the 1,000th dimension, we can connect even better and closely understand how related things are in the spheres of knowledge. And I won't go further than that. And that's just the fundamental of large language models, the powers of ChatGPT and artificial intelligence age that we actually look at now. Ability to actually see things in a greater dimensions and the hands of artificial intelligence, which are a form of deep neural networks where we can actually connect things together within a multi-dimensional space beyond which our human mind can actually reason, but we can conceptualize and actually understand. So we see artificial intelligence playing a more critical role in pushing us forward and being able to actually address disease processes and also augmented clinical decision-making with algorithms that are capable to identify patterns that we can never have seen with our ordinary eye. And so where we're going from here, we're seeing personalized molecular blueprints became the mainstay, where I tell you earlier on, you can have your whole genome sequence and we can tell all the molecular changes in your entire genome, but disease states now. But then beyond that, we can even see much more than that. We need to supercharge our intelligence to be able to make use of that information. So ability to supercharge that intelligence to be able to make more precise decisions is where we're going. And then, like I said earlier on, I said there is no true demarcation between philosophy and science. We have over time defined physical science, biological science, chemical science. But at a very, very fundamental basis of understanding, there is no wall between knowledge. There is no wall between chemistry and biology. There is no wall between physics and medicine. And in fact, it would amaze you that in the last 2 years, the Nobel Prize in medicine and philosophy and physiology was actually won by computer science, by AI. It was people that developed AI, artificial intelligence, and made it to actually look at AlphaFold, and AlphaFold explain protein structures. Because your proteins, you think, yeah, it's biology. But if you look at the molecular arrangement of 2-dimensional, 3-dimensional structures of proteins, it's a problem that cannot be solved by just biologically. You have to think in a physical science way, how the molecules and atoms arrange themselves in 3-dimensional space. And we can figure it out by just ordinary means. But using artificial intelligence to solve that actually makes it possible for us to answer what is the 2- or 3-dimensional nature of proteins and how do they function in human beings. So breaking down institutional silos and the ways we imagine knowledge, that's something we'll be seeing from here on. And I'll close with this quote from Eliud Kipchoge, as one of the greatest marathoners of all time. Some of you might know him. It said the best time to plant a tree was 25 years ago. It said the second and the best time is today. You plant a tree of self-discipline. whatever future you can imagine, whatever you think tomorrow could become, you have a contract to sign with yourself to be disciplined enough to take the step today to get to that future. So are you still looking at the horizon for what the future is, or are you starting to take that steps towards that future? I think we'll get to questions and reflections from here. Thank you.
Okay. Thank you so much, sir. Good evening, everyone. Please, if we can hear me clearly, I'd love to give our speaker a virtual round of applause, please. Or we type in the chat box that thank you, sir, because that was a really amazing session. That was an amazing session. We've learned so much. We've learned that medicine is now evolving rapidly, like at a very, very fast rate, and that we need to be able to keep up with the recent digital tools such as artificial intelligence if we want to truly evolve as a medical or clinical practitioner. Thank you so much, sir. So right about now, when I started the session, I said we're going to be having a very important figure. So we introduced Dr. Paul Aiyetan, and he had the session. And now I'm going to be introducing another special figure. She is a mother to us all, and she is the founder of the FAJIM Medicare Foundation. I'd love to invite Professor Fatima Abdul Karim to give a founder's remark. Thank you so much.
Wow, wow, wow. Thank you, Dr. Paul. I was so carried away that it felt like you should just continue, dear scholars. I don't know whether you been able to get who Dr. Paul is. This is someone who finished medical school, just like you're in medical school now. And all you're thinking is, oh, I want to do residency, I want to go to OD, I want to do pediatrics, I want to do this, I want to do that. But look at the path that he took. Went to John Hopkins because he's someone with a critical mind, someone who likes to ask questions, who wants questions answered, not just asking questions, but also want to be part of, the solution. So he went to do, combination of engineering, medicine, philosophy. Do you get that? And where is medicine going now? Medicine is going to or is at a stage where it is not just enough to say, as pathologists now, we don't just end up and say, oh, and name the disease, give the disease a name. We go to the extent of, saying, yeah, this is the disease name, but we also want to be able to tell the patient what the patient will benefit from. Because, for instance, look at breast cancer. Breast cancer in Mrs. A is not the same as breast cancer in Mrs. B. Look at colorectal cancer. Colorectal cancer in Mr. A is not the same, even if they are born by, even if Mr. A gave birth to Mr. B, because they are different. The molecular types are different. Now they are talking at the level of the cell, at the level of the proteins, at the level of the minute, the minute details. And with the humongous amount of data that is now available, what his company does is to have everything under one roof, right?
Correct.
To have the clinical information, to have the pathology, the morphology, right? And then to have the molecular information as well. And now getting all this data together and using AI to, AI algorithm to be able to say, oh, this is exactly what the patient has. This is what the patient is going to require to treat that patient. Okay? So where I'm going is, do not limit yourself. And when we are talking, from that quote that he gave at the beginning, that where you are going, let me look for it again, that the future that you are looking at, it is not the one that you're going to keep going and then you find it somewhere, right? It's the one that you're going to create by yourself, that you're going to make. And making that future starts now. And you should stop thinking like, our traditional ways of thinking that, there are so many diverse ways, diverse paths that you can take to get into that future. Okay? The future is not some place that you're going, but the one you are creating. You're creating that future by all that you're doing. And so you should begin to think, have a critical mind. Where do I want to be? Where do I want to see myself, in that future that you're looking at? And how do I want to go there? And it doesn't have to be, one traditional way of, oh, okay, you finish, then you do residency, then you do PLAB, then you do this, then you do that. There are several other paths that you can take to get into where you're going. So they say think outside the box, even in creating your future. But now they say chatter the box. Dr. Aiyetan chattered the box.
Thank you.
He chattered the box.
Well put.
I mean, going to doing engineering and, and see where it is now. So I want to encourage you that this kind of lecture is not a lecture. Even if you go reading, those of you that have missed this lecture, even if you go reading through the slides, it's not the slides, but the lecture is supposed to sort of inspire you so that you also think of how do you get to that future? How do you begin to create that picture, to create that future? And I like when it was talking about we like to talk about idiosyncratic reaction in patients. And then when we want to talk about etiology of diseases, we will say, at the end of listing everything, we will say some are idiopathic, isn't it?
Correct.
Idiopathic, because we don't know. But many of those things that we did not know then, I remember when you talk about viral hepatitis, in my school days, we used to have only hepatitis A and B, and then others, non-A, non-B. Now we have more than 10, and the number is increasing.
Correct.
Okay? So, have a broad view of life in whichever specialty whichever course that you're reading now. There are a whole lot of things that can be done. There were those days that say, oh, okay, because you're doing dentistry, you cannot, if you, like I have, if one of my students then who finished dentistry and wanted to go into public health, they said, no, she couldn't go and do public health. Now she did that public health, and then now she did mass communication, And now she's into health communication. How do you, talk to, how do you interpret or tell the story of the outcome of research in the language that the common man will understand? So there are several paths that you can take to get into that future. Begin to plant the tree of self-discipline, as he quoted that, is it that Ethiopian?
Yeah.
So is it self-discipline, or whatever that you have decided to do, and you will see yourself there in the nearest future. I want to thank you again, because I'm sure you have inspired a scholar. I'm also inspired myself, and I'm really, really very interested in not just pursuing our interaction further with in line with our scholars, how we can even go about, if anyone is interested in, some kind of internship or exposure for brief period. I'm sure we can always organize such, And my area of research sort of aligns somewhat with what you're doing. Currently we started the colorectal cancer clinical trial for MSI MSI colorectal cancer. Yes, we're using immunotherapy for them. So it's I'm sure there is a lot that we can gain from you in future. Thank you once again. Thank you very much. And thank you, Mariam, for getting Dr. Aiyetan for us. So over to the anchor. I hope I've not taken too much of your time.
No, ma'am.
No, not at all, ma'am. Okay. Thank you so much to our mother and the founder of FAJIM Medicare Foundation for that amazing remark. I'd love to go back to something she said, and that was, anyone who's not in this call right now may not really be inspired. So those of us that are on this call, we scholars are on this call, we 99% or even 100% sure that we are inspired, and I'd love to testify to that fact. I have been inspired. Yes, I've been really, really inspired, because I'm a medical student and I still don't even know what I want to do. But listening to Dr. Aiyetan's session now, I think I want to think more outside the box and diversify. So, in view of this, I would love to thank Dr. Aiyetan once again for sharing his insights and his vast knowledge. Thank you for letting us see that there's so much that we can actually do, and we should not confine or restrict ourselves to just one perspective, because like a quote was mentioned, that the future is not some place where we are going, but the one we are creating. Thank you so much, Dr. Aiyetan, for making us see that reality. Okay, so right about now, we're going to be having an interactive Q&A session. So to my fellow scholars on the call, if we have any questions, please, this is the time for us to ask our questions. So if you'd love to ask your question verbally, you can just raise your hands. But if you'd like to type, just type in the chat box and I'll be reading the questions out. Thank you. So please, let's have our questions now. We can type our questions in the chat box if we do not want to speak.
I see someone's hands up.
Okay.
Is it Dr. Ganiyu? Dr. Ganiyu Damilala or Damilare?
Yes,
Yeah,
sir.
I can see that. Okay, yeah. Please go
Good
on.
afternoon, sir.
Good
Good afternoon, everybody. I'm really grateful to be here, and thank you for having this session this morning. I feel like So I would have beat the session, I would have been so quick because this is like a very crucial time to have this like session. So I've been super confused, been especially
Hey,
using AI
I think
in
there's a
being
problem with your mic.
Adi, you
Hello,
are
I think there's a problem with your mic.
there is some
Is it better
echo.
now? Is it better now?
Yeah, better.
Okay. Sorry, ma'am. Sorry, sir. So, good afternoon, sir. Good afternoon, Prof. Good afternoon, everybody. So, I'm really excited to be here, and I feel like if I would have missed this session, I would have been so pained because I feel like this really answers a lot of my question and also really makes me like want to ask more questions. So, like I'm really grateful that you're taking us this session, sir. So yeah, as someone that is actually interested in melanoma research and using AI and machine learning algorithms to actually foster early detection of melanoma in African communities, especially because in Nigeria we have like fewer dermatologists, and I'm always like looking forward to like maybe use like AI models to like train Nigeria skin tone so that we'll be able to like diagnose this faster and make like the process really faster. So your talks really kind of like make it make sense to me. So my question is that like in med school, how did you really plan out this your pathway of med school? Because I've really been interested in after med school, like taking a PhD. Like, did you do your house job? Did you do your residency? Or did you just finish med school and the next thing, masters at John Hopkins and cancer stuff like that? So I want to really know how you actually processed the journey from med school to John Hopkins and then the rest. And also, like Prof said, like it would be really nice if we can have like probably an internship or like a program where, if possible, we can learn on that because I would really like to learn a lot, especially because you really took us down to the molecular level and really explained everything so much on how we can actually take our disease decision making down to the molecular levels, like in to like improve patient outcomes. So I really like you can answer those questions, sir. Thank you very much, sir.
You're very welcome. Thank you. Thank you very much for asking that question. Yeah, so I will tell you my journey. And I think oftentimes we we don't see what we have around us. And I would tell you categorically for me, um, I credit my journey to the people I had around me. Um, when I remember people like Dr. Happy, who is currently a professor at Redeemer University, uh, Professor Mayo Wawalabi, who used to be, it was my senior reg in neurology, senior register in neurology. That's currently a professor at University of Ibadan. Professor Guni, Professor Akin, um, Professor Motade, and these are people that were close to me and that knew me, and I asked questions. I will have these questions in my mind, and I pose it to them, and I ask them in a respectable way. And seriously, when you ask those questions in a sincere way, your teachers will see through that you truly are genuinely interested in learning. And here is what happened. Professor Motade connected me with Dr. Happy, who had recently come back from Johns Hopkins and had a funding to set up a molecular biology lab at Institute of Advanced Medical Research and Training. And because I kept asking questions about molecular processes, how can we understand these processes? Like you're asking melanoma, how can we use AI to better understand the skin pathology that is typical of Africa, that in African is so different, somewhat different from the way it is in Caucasians? Those are genuine questions. We start from a clinical curiosity, and that my slide, start from a clinical curiosity. And so connected with Dr. Happy. And then I would spend my evenings in the molecular biology lab when everybody else is going to bed or going to play soccer. And after medical school, Professor Gunayi and these people just put a recommendation letter for me in my application to Johns Hopkins University. Because then we look for, okay, this is what I'm interested in. This is what I think the future is. The future I was imagining. And I found the schools and I said, I need a recommendation. And they're so delighted to write a recommendation for me. In fact, Dr. Professor Owolabi right now, at that time, connected me with the professor at Johns Hopkins University that he had met at a conference. So maximize the resources you have available to you. And the moment they see your genuineness in pursuing knowledge, they begin to open up to you and connect you to where the next opportunity is for your growth. And the same thing happened at Johns Hopkins University. The moment I got to Johns Hopkins University, Mon PI Luigi Marchioni had just come from, he had just been recruited from Europe to Johns Hopkins University, and he had just completed his MD PhD program. And it was starting in my local biology lab, and he was asking the same question I was asking. So come on to my lab and let's do this stuff together. And then he connected me with Sarah Whelan, who connected me to the funding that NSA had provided in the lab of Professor Chan. So maximize the resources that you have and embrace them and ask pointed questions. And sincere educators, when they can answer your questions, they're pointing to where they think you have the opportunity to learn. So that was the way I started. And when I get to Johns Hopkins, it's a entirely different world. It's like, now you don't have, in fact, Luigi Machunas says there is no textbook for us to do this work. But then we know we need this knowledge, and that knowledge. And then I go to the advanced physics lab, which is where the atomic bomb was actually created, I got to be there. And because that's where the people that have the resources and knowledge for the questions you're asking and the solution you're trying to create, actually deeper side. Ask the questions, be genuine, be open, and do not back out. And don't feel you have the wall against you. The world is open. There are countless and many opportunities and sincere people that really want to see people coming behind, really go ahead and even do better and much more than they've done. Did I answer your question?
Yes, sir. Thank you very much, sir.
Okay, thank you so much, sir, for that wonderful insights. Okay, so is there anyone else with the question? You can go on, please. Okay, so someone here actually had a question, but I think it was related to the first question asked. So she says you have answered that one as well, sir. So anyone else with a question, please, we can type it in the chat box.
Hi, I just have a quick comment. This is Dr. Adam. Dr. Paul.
Hey, Dr. Adam.
Very inspiring talk.
Thank you.
Yeah.
Good to know you're there.
Yeah, and it's nice what you say. I think what I just wanted to say, two things just piqued my mind. The quotes that you gave, there's a quote that also kind of aligns with what you have said. It's one by Rumi that says the way is made by walking. That you make the path just as you walk along it. Essentially, that's what it means. So people have thought about life and how to make life, and essentially, you only get to make life by doing it. So thank you so much for the talk. The other thing I wanted to say is these things are actually real life. Someone mentioned melanoma, discovering its genetics. And just recently, I think that there's a phase 3 trial for Moderna vaccine. And I don't know if you're aware that that's just been approved. Yeah. So, and just because of that, there's like an 80% increase in the stock price of Moderna. So these things, it's not just the books, it's actually business, it's money. We also, just as we're thinking about the questions, we also have to be conscious about the business side of things, the application, because you have to marry all of that to make your way. So thank you again for the talk. Thank you so much. Bye.
You're welcome. Well, thank you too. And thank you for facilitating this talk as well too. And I would just add one more to that comment. This is something I kind of realized that value always, I said money follows value. And as young people, we are at a very opportune moment in life where we have the strength and the capacity to pursue after value and build capacity. And the reality is this as the value will follow. And so, yeah, Dr. Rahman talked about money and the commercial side of things, which is a reality. And people will pay for what they know the derived value from. And people would pay utmost price for what they think is a high and highly significant value. And so as you think about the future, you could think about it in the sense that where do I see the most value? Where do I see what persons and people would benefit from? Where do I see that once this solution could be arrived at, we could solve a whole lot of human problem. Remember, the more the problem we can solve, the more people we can solve it for, the more critical that problem is to life, the higher the values. And the higher the value, the higher the return and the benefits that accrue from such value. And so it's a laudable pursuit and career aspiration. When you discover and begin to reason and think about that, as you journey through life and discovering career.
Okay, thank you so much, sir. Thank you, Dr. Adam, for the wonderful contribution and comments. So, again, do we have any questions to my fellow scholars? Do we have any other questions for Dr. Aiyetan? Okay, I think there's something here in the chat box. So Adewale Nuseba said, thank you so much, sir. This is so insightful. Just as Prof said, we would really like to get an internship. So the person said she's now more interested in research. I think that was just a comment. Okay, but she said she's now more interested in research. That was from Adewale Nuseba.
Yeah, we would be working with something like that. We'll be working on that with Prof after this talk. And I think Dr. Kudus Ibrahim has his hand up too.
Okay, please, you can go on. You can go on. Yes.
Dr. Kudus, you could go ahead and talk.
Good evening, sir. Good evening, Prof. Good evening, everyone. Thank you, Dr. Paul, for the insightful session. I'm currently a fifth-year PharmD student at the University of Ibadan, and I want to ask a question concerning pharmacogenetics. So recently, in one of our lectures, we did presentations on different pharmacogenetics issues, and there was a particular one that caught my attention concerning warfarin dosing in Nigerian population.
What do you call it?
There's a particular pharmacogenetic issue concerning warfarin dosing.
Okay, warfarin dosing.
The dosing of warfarin yes, in Nigerian population. So there's this particular enzyme called DVCOC1. I'm very sure you know about it. That the warfarin blocks to prevent the conversion of the So the data that is used for the warfarin dosing in Nigerian population was extrapolated from the research done in Caucasians and Asians, and it does not exactly apply to Nigerian population because Nigerians, they are very genetically, Africans generally, blacks, they are genetically more diverse than whites and Caucasians. So my question is that how can blacks, Nigeria, and maybe Africa at large, I know we are currently going towards precision medicine, pharmacogenetics, but yeah, we have still not gotten there yet compared to the developed nations. So, as pharmacy students, medical students, and students in the field of medicine, how can we, if we are interested in things like this, maybe conducting research on, for example, this Vicoquan warfarin that affects warfarin dosing, so that we can get data that is specific to the black population.
That's
I
a
don't know if
really,
I'm able
really,
to frame
really
the question
fantastic and
properly.
amazing questions. And you're asking, are you done asking the question before I start asking?
Yes, sir. Yes, sir.
And you are asking really germane questions that everybody in the biomedical and scientific field is troubled about with respect to precision medicine, the future of it, and the African subpopulation. Because despite the fact that we are so diverse in population, we do represent just about 1% of the entire data used to make inference about what constitutes molecular changes and what is specific genetically, particularly with respect to disease. In fact, in the biomedical precision medicine space, the data used there does not have much of African representation. And that's something I think we need to start working towards and the problem, it's not so far-fetched because the cost of profiling and running these experiments are still prohibitively large to be shouldered by small institutes or institutes that are not well funded. Okay. And so as the many prongs the many ways you could look at it and start to think about it. And I'll mention one other approach that I'm working with a group about. We have factors that goes all the way from cost to even cold chain issues. As some of these experiments require reagents that need to be frozen for really long and prolonged period of time. And so it's not only the cost, we have logistical issues that actually make these experiments really complicated and complex. Not that they can be done, but these are things we need to address to make many of these possible locally. And I'll tell you, when I said earlier on, I will go to the IMRAT. You know IMRAT at UCH, right? And I'll go to that lab and I'll be running the PCR experiments. And if by mistake, I'm in an ABH and the light goes off and my PCR experiment is running, I have to run back and make sure the generator comes on. Otherwise, the entire reagent and experiment is all wasted. 3 hours and up to 6 hours of work, all wasted. So we had those issues back then. I think it should be less of an issue right now. But how can we participate? First of all, we know we have this problem, we have these questions, we need it solved and answered. And how can we make it happen? Find a way to encourage and canvass for additional funding for these kind of experiments and studies to be done in the local population so we can base finding and approaches to what we have in the local context. 2, develop alternative methods for profiling where the cost of running experiments are not as expensive. One of those that we engage with a group is using saliva diagnostics, where we're beginning to look at what is called exosomes in saliva. The saliva that's produced, there are cells in the mouth that do release what's called exosomes. Exosomes are some capsular bodies, as in like micellar bodies, where you have some genetic content within them. They're releasing the saliva, and we're profiling them to see whether we can correlate what we find molecularly in those exosomes and even in the saliva and the cells that are shed in the mouth into the saliva to what really constitutes the genetic makeup of persons. The good thing about that approach is that it doesn't require as much a cold chain as a typical in classical or molecular experimental studies. If we can do that and we could correlate to what is happening in pathological processes, it's an added advantage. So what can we do? First, converse for funding. Two, find alternate means, like go to some other lab and direct approaches to overcome these challenges that are actually pose as a problem to making those studies possible locally. Three, develop modalities and methods and approaches. And these are other areas of research that you can also think about. How can I make these processes or these experiments possible given the limitations I have in my environment? So for PhD research, I just give an idea. How can we do high throughput research given the limitations that we have in the locality? So those are ways you can begin to think about it, but never stop asking the question. But don't stop at asking the question, move one step ahead and saying, how can I solve or resolve the question I ask in spite of my limitations or above and beyond my limitations? Does that answer your question?
Yes, sir. Thank you very much. Thank you so much.
Okay. Thank you, sir, for that wonderful insight again. Okay, so everyone, because of time, we are going to be wrapping up the Q&A session right about now. So as we come to the end of this wonderful and insightful session this evening, I'd love to hand over to another fellow scholar of mine, Hadi, who's going to be delivering the vote of thanks. So Hadi, over to you now.
Thank you very much. It's a privilege to be here, and yeah, I'll first really like to appreciate Dr. Paul Aiyetan for speaking with us and taking really part of his precious time to show us his progression from med school to postgraduate school and what he's currently doing in precision medicine and artificial intelligence. It's really insightful. Personally, if I wouldn't have attended this session, I would have been so pained and a lot. And I believe other scholars are so much appreciating that as well. And I also like to appreciate Prof. Abdul Karim. She has really been taking us and bringing really people that are really doing great things in their field, in healthcare, in corporate world. She's been really that mother to us, and I pray that may God continue to make her reach greater heights. And yeah, she keeps raising the bar. I also like to appreciate Miss Miriam Rahman for coordinating this session and making sure that we are here on time, and also to our moderator. I appreciate you as well. And then to all the FAJIM scholars, we want to say that we really appreciate and we learn a lot from this. And yeah, we really hope to be really conscientious and impactful healthcare leaders that really transform healthcare outcomes in Nigeria, Africa, and in the world. Thank you, everybody.
Thank you too. Thank you too.
Okay, very quick one, sir. Thank you so much, Hadi, for that. Sir, is it possible that with the scholars, is it possible that we connect with you perhaps
Absolutely.
via any of your social media handles?
Please do. I really would love to. Send me an email. Talk to me anytime. And Miss Miriam, please feel free to share my contact information with the scholars. I really appreciate it. I really love to hear from them. Yeah.
Thank you.
Mm-hmm. And you find me on
Okay.
LinkedIn, go ahead and connect with me.
Yeah, okay. Thank you so much, sir. Thank you
You're
for the opportunity. Okay. Miss Mariam, I don't know if there's anything else before we formally close this session.
No, I think that would be all. Thank you very much, Toiba.
Okay, thank you so much, everyone. So please let's enjoy the rest of our evening. Good evening once again, everyone. Good evening.
Thank you, everybody.
Thank you so much. Thank you.
Thank you.
Bye.
So, bye.
Okay, sorry, everyone.
before our speaker leaves, can we just unmute our mics and say thank you so much, Dr. Aiyetan, all together in the chorus.
Thank you.
Thank you so
So
much,
much.
Dr.
So much,
Aiyetan.
Dr.
Oh,
Aiyetan.
Aiyetan.
Thank you,
Thank you
we're
for
sir.
really
having me.
grateful for this amazing session.
Thank you for having me too.
Thank you.
Bye.
Bye.
Okay. Bye, everyone.
Yeah, bye, ma. Thank you so much.
Yeah.